I hope you all are not getting stir crazy yet, since it's only the first week or two of class. But just in case you are, I thought I'd put a funny on here to give you a good laugh. I must agree that this is by far the most accurate graph I've seen during the pandemic. With that aside, let's move on to talking about multiple sclerosis drug action. I must warn you that this video is a little bit longer than the last two since there are several drugs that I have to cover. But we will get through all of them together. Enjoy! Let's quickly recap the most important aspects of the pathophysiology of MS. MS is a cell-mediated immune response that involves the initial activation of CD4 positive Th1 helper T cells that then migrate and infiltrate into the CNS from the periphery. T cells activate macrophage and CD8 positive killer T cells that then lead to damage of myelin as well as damage to oligodendrocyte cells themselves. However, as a reminder, remyelination can occur if this process is stopped early enough. And that is the premise of the disease-modifying therapies, or DMTs, that we'll cover in this section. Here is shown a broad overview of the MS therapeutics that we will cover. Again, there are many drugs to go through, and unfortunately, many of them have quite different mechanisms of action because they are drawn from cancer therapy, immune therapy, as well as other areas. The therapeutics are broadly divided into three areas: drugs that are used to treat acute exacerbations, and there's really only one drug for that, which is methylprednisolone. Drugs that are used as disease-modifying therapy that are administered chronically and continuously over a long period of time in an attempt to slow and or stop the disease process. And finally, drugs that are used to treat secondary symptoms, such as muscle spasticity and incontinence. These are consequences of MS damage that can be treated by drugs that have really no effect on the underlying pathophysiology, but are treating the secondary symptom of the disease. And I'll really only highlight one in this section that's used for muscle spasticity. Other drugs for secondary symptoms you either have learned about already or will learn about shortly. Methylprednisolone is indicated for MS acute exacerbations only. Methylprednisolone is a systemic corticosteroid. It is a glucocorticoid specifically, and can be administered via either oral or IV administration during an MS acute exacerbation. Methylprednisolone is not considered a DMT and has no effect on the underlying disease progression. Methylprednisolone is considered an intermediate potency corticosteroid. Its half-life is on the order of a half an hour to 3 hours, depending on whether it's administered oral or IV, which makes it an intermediate acting corticosteroid. Its mechanism in ameliorating an acute exacerbation of MS is that it reduces the migration of polymorphonuclear cells, these are things like neutrophils and basophils, into the CNS or into sites of injury. And it also decreases capillary leakage. Now, capillary leakage is the mechanism that allows these immune cells to get into the CNS in the first place. So by reducing polymorphonuclear leukocyte migration and capillary leakage, you wind up with fewer immune cells in the central nervous system that are able to cause damage. A few adverse drug effects or adverse drug reaction, ADE and ADRs, to watch out for is that this drug is immunosuppressive, and combination or administration with other immunosuppressant drugs should be avoided. Methylprednisolone is a substrate of the cytochrome P450 enzyme 3A4, it is metabolized by CYP3A4 to inactive metabolites. And so co-administration with drugs that either induce or inhibit CYP3A4 can affect blood levels of methylprednisolone. And care should be taken when administering methylprednisolone with any drug that is a CYP3A4 inducer or inhibitor. Next, we'll move on to talking about the disease-modifying therapies, or DMTs. Again, just to remind you, these are drugs that are able to at least somewhat modify the underlying disease course and hopefully slow the pathophysiology of the disease. The first group of drugs I will talk about are the interferon beta drugs. Interferons are a group of endogenous cytokines that have very diverse modulatory effects on the immune system. Interferon beta is a synthetic version of endogenous interferon that differs from the endogenous protein by only one single amino acid. Its mechanism of action in MS is to reduce antigen presentation to T cells, and it also reduces T cell infiltration into the CNS. By doing this, the result is fewer activated T cells in the body in general, and also fewer activated T cells in the brain and spinal cord. There are some adverse effects to note with the administration of interferon beta, the most common ones being flu-like symptoms, runny nose, cough, etc., as well as injection site reactions. There are some noted neuropsychiatric effects, things like depression, but those are a little bit less frequent than either of the other adverse effects. There are several different interferon beta products available, and they differ in a few key aspects, including route of administration and half-life. The first I will talk about is Avonex, which is interferon beta 1A. This is administered by intramuscular injection and has one of the shorter half-lives at about 19 hours. Rebif is an interferon beta 1A product as well, but in this case is administered by subcutaneous injection and has a relatively long half-life at 69 hours. Plegridy is also an interferon beta 1A product, but it contains an additional functional group called PEG, or polyethylene glycol. This pegylation results in increased absorption and distribution throughout the body. It is administered by subcutaneous injection and has a relatively long half-life at 78 hours. Finally, beta-serin is an interferon beta 1B product that is also administered by subcutaneous injection and has the shortest half-life at 4 hours. The interferon beta products are used for relapsing forms of MS, and they are considered to have moderate efficacy overall when compared with other drugs. The next two agents are considered cytotoxic drugs. These are agents that are actually borrowed from the cancer field, and they have cytotoxic effects on immune cells, given that immune cells are a type of rapidly dividing cell. Because of that, they also have significant side effects. The first of these cytotoxic agents is teraflunamide. It is a member of the class of pyrimidine synthesis inhibitors. And specifically, it is inhibiting the rate-limiting enzyme in the synthesis of pyrimidine bases. And because this pathway is very important for the function, activity, and activation of B cells, the net effect of teraflunamide is a reduction in B cell activation. This decrease in B cell activation means less antibody production and less damage to myelin. It has an extremely long half-life of about 18 days, and this is thought to be due to enterohepatic recycling. Teraflunamide is a substrate of several metabolic enzymes and transporters, and therefore has the potential for significant drug-drug interactions. And so a database of drug interactions should be consulted whenever teraflunamide is administered. The most important adverse effect of teraflunamide is that it produces teratogenic and embryo-lethal effects. It can cause damage to the fetus and in some cases even lethality, and should be avoided at all costs in women who are attempting to become pregnant or who are pregnant. Another adverse effect that is common is hepatotoxicity. Teraflunamide is also indicated for relapsing forms of MS. It is administered orally and also considered to have moderate efficacy. Cladarabine is the other of the two cytotoxic drugs. It is also used in cancer chemotherapy. Cladarabine is a purine antimetabolite. So where teraflunamide reduced pyrimidine synthesis, cladarabine decreases purine synthesis. Its mechanism of action is that it is actually itself incorporated into DNA. This causes DNA double-strand breaks and problems with DNA repair. Again, because T and B cells are both rapidly dividing cell types, they are more susceptible to this damage caused by cladarabine. And therefore, you have reduced T and B cell activation in individuals with MS. One important thing to note about cladarabine is that it is a prodrug. It is taken up into the body and then actually taken up inside of cells where it is activated to the active drug by phosphorylation. It is a substrate of many different drug efflux transporters, and therefore, again, a database of drug interactions should be consulted whenever cladarabine is used. As a cytotoxic drug, it is also known to have teratogenic and embryo lethality effects. Because it is also immunosuppressive, it can lead to one of the two most common side effects associated with the use of immunosuppressive drugs in general, and that is the development of a secondary or opportunistic infection or the development of a new onset malignancy or tumor. It is indicated for relapsing forms of MS only where an inadequate response to other therapies has been observed. It can be administered orally and is also considered moderate efficacy agent. The next set of drugs I will cover are miscellaneous agents that all act as disease-modifying therapies, or DMTs. The first of these is glutamate acetate, which is classified as a biologic drug. It is actually a mixture of random polypeptides, random protein sequences that are comprised of 4 different amino acids only. And these are lysine, glutamic acid, alanine, and tyrosine. The mechanism of action in MS is not exactly known, but it is thought to act by decreasing antigen response to myelin proteins. So the mixture of random polypeptides comprised of these 4 amino acids actually activates regulatory T cells or suppressor T cells that are able to dampen the immune response to the myelin proteins we mentioned earlier: proteolipid protein, myelin basic protein, and myelin oligodendrocyte glycoprotein. Suppressor T cells then reduce T cell activation and lead to a decrease in damage directed against those myelin proteins. The most common adverse effects associated with glutamate acetate are injection site reactions and chest pain. It's indicated for use in relapsing forms of MS as an IV drug, and it is also considered to have moderate efficacy. The next group of drugs I will talk about are all part of the family of sphingosine 1 phosphate receptor modulators. I'm going to talk about the mechanism of action of these drugs here first before I go on to then compare the different S1PR modulator drugs available. The main effect of these agents is to decrease T cell release from lymph nodes, thereby decreasing the number of T cells that are available in circulation. Sphingosine 1 phosphate receptor modulators do this by blocking the exit of T cells from lymph nodes. As you can see in the figure on the left, in the absence of drug, sphingosine 1-phosphate, the endogenous ligand for these receptors, contributes to the normal process of T cell exit from the lymph node. When these modulator drugs are on board, the sphingosine 1-phosphate receptor is absent or missing from the surface of T cells, and therefore these T cells do not have their normal signal to be able to exit the lymph node, and they are sequestered there, and they cannot eventually migrate to the CNS and cause damage. There are 3 S1PR modulators currently on the market. These include fingolimod, siponimod, and ozanimod. Again, they are all sphingosine 1-phosphate receptor modulators. The primary difference in their mechanism is that fingolimod is non-selective for multiple sphingosine 1-phosphate receptors, whereas both siponimod and ozanimod are more selective for sphingosine 1-phosphate receptor subtypes 1 and 5. This is thought to reduce some of the adverse effects that are associated with the use of fingolimod. Their mechanism is all the same, as we just discussed. It prevents lymphocyte or T cell exit from the lymph node, and therefore fewer T cells in circulation. Fingolimod is a prodrug and is activated by cytochrome P450 3F4, and therefore drugs that either induce or inhibit CYP3F4 should be carefully monitored in patients. However, given that 3F4 is a more minor CYP, this is not a concern with too many other drugs on the market. Siponimod and ozanimod do not require activation, and they also have a much shorter half-life than fingolimod. Ozanimod is interesting because the primary action is actually due to some of its activated metabolites as opposed to the parent drug, even though the parent drug does have activity itself. Siponimod is subject to interactions via the cytochrome P450 2C9 enzyme, and therefore, again, drug interaction and in this case, polymorphism data should be consulted whenever siponimod is administered. Ozanimod does serve as a substrate of several P450 enzymes as well as drug efflux transporters, and therefore a drug interaction database should be consulted with use of this drug as well. Now, the primary adverse effect associated with the use of fingolimod is cardiotoxicity and to a lesser extent macular edema. Use of fingolimod is associated with AV block, and this toxicity is often problematic related to its use. This is where the newer agents, siponimod and ozanimod, come in, being more selective for S1PR subtypes 1 and 5. Siponimod was shown in clinical trials to have less cardiotoxicity than fingolimod, and ozanimod really has little to none. Another thing to note is that there have been cases of progressive multifocal leukoencephalopathy with fingolimod, or PML. Progressive multifocal leukoencephalopathy is the result of an opportunistic infection in the brain with the John Cunningham virus, or JCV. The primary risk factor for developing PML is lymphopenia, which is a reduction in the number of lymphocytes. And so often monitoring lymphocytes is a good way to determine whether someone is at higher risk of developing PML. The primary sign of PML is progressive asymmetric muscle weakness. And in order to determine whether someone is susceptible to developing PML, patients are monitored using complete blood count and also MRI. The S1PR modulators are indicated for use in relapsing forms of MS. They are administered orally, and these are considered high efficacy DMTs for MS. The next group of DMTs I will talk about are the fumaric acid derivatives. There are 3 drugs on the market that all fall under this class: dimethyl fumarate, or DMF, diroximel fumarate, or DRF, and monomethyl fumarate, or MMF. Again, these are all fumaric acid derivatives. Their mechanism in MS is also complicated. They are thought to decrease oxidative stress and inflammation in the brain by activating the NRF2 transcriptional program, which is a known anti-inflammatory pathway. Dimethylfumarate and diroximel fumarate are both prodrugs. They are converted to the active substance monomethyl fumarate once inside the body by esterases. In contrast, monomethyl fumarate is already in its active form, and it is metabolized to inactive substances by the TCA cycle. These agents do not have significant drug-drug interactions, although alcohol may decrease serum levels of diroximel fumarate specifically. The main difference between the three drugs is the way in which they are metabolized. Dimethyl fumarate is metabolized to the active drug monomethyl fumarate with the formation of methanol. One of the reasons that this drug can cause hepatotoxicity and GI side effects is because of the formation of high quantities of this methanol metabolite. In contrast, diroximel fumarate is primarily metabolized to monomethyl fumarate and an inactive metabolite that is not damaging, whereas a more minor metabolic pathway will wind up forming the methanol metabolite. And therefore, diroximel fumarate is thought to have fewer side effects. Monomethyl fumarate is also thought to have fewer side effects given that it is already the active drug and does not need to be metabolized for activation. In general, the primary adverse effects of these agents are hepatotoxicity, occurrence of opportunistic infection, including John Cunningham virus, which can lead to progressive multifocal leukoencephalopathy. And as I said, in general, diroximel has fewer side effects, including GI side effects like nausea and vomiting, compared to the other two agents. These drugs are indicated for relapsing forms of multiple sclerosis. They are administered orally and are considered to have moderate to high efficacy. The next three agents on the list are all monoclonal antibodies and are relatively newer drugs that are used as DMTs in multiple sclerosis. The first antibody I'll talk about is natalizumab. This is a humanized monoclonal antibody directed against the integrin protein VLA4. VLA4 is a protein found on the surface of T cells that is integral for T cell adhesion, migration, and infiltration into the CNS. By inhibiting this protein, natalizumab decreases T cell adhesion and infiltration. Natalizumab is highly immunosuppressive, and therefore administration with other immunosuppressants should be avoided. Its primary adverse effects are the development of opportunistic infections, and this also includes John Cunningham virus, which can lead to the development of progressive multifocal leukoencephalopathy. Hypersensitivity reactions are also possible with administration of natalizumab. One of the pharmacokinetic aspects to look out for with natalizumab and other monoclonal antibodies is the formation of anti-drug antibodies. These actually increase clearance of natalizumab from the body and can decrease the effectiveness of the drug over time. Natalizumab is indicated for relapsing forms of MS. It is administered via IV and is considered a high efficacy DMT. The next monoclonal antibody on the list is alemtuzumab. This is again a humanized monoclonal antibody that is directed against the CD52 protein. CD52 is a protein that is found on the surface of both B and T cells. And the alemtuzumab binding to CD52 results in the depletion of both B and T cells by antibody-dependent cellular cytotoxicity, or ADCC. The premise of ADCC is that these cells that have been bound by alemtuzumab are decorated with antibodies that target these cells or direct these cells for cellular cytotoxicity by various mechanisms. As with many monoclonal antibodies, the half-life is very long. For alemtuzumab, it is on the order of 2 weeks. Alemtuzumab is an immunosuppressant, and administration with other immunosuppressant drugs should be avoided. Its primary adverse effects associated with use are the presence of a secondary autoimmune disease, infusion reaction, development of a secondary malignancy or tumor, and stroke. Alemtuzumab is indicated for relapsing forms of MS with inadequate response to a previous therapy. It is administered IV and is considered a high efficacy DMT. The last monoclonal antibody indicated for use in MS is ocrelizumab. Ocrelizumab is a monoclonal antibody humanized that is directed against the CD20 cell surface protein. CD20 is a cell surface protein that is specific to B cells, and therefore ocrelizumab depletes B cells from circulation by both antibody-dependent cellular cytotoxicity, or ADCC, and the activation of the complement system. Ocrelizumab has some interesting pharmacokinetics. Its onset of action is about 14 days, and its duration is on the order of 72 weeks. Again, like other monoclonal antibodies, ocrelizumab is immunosuppressing and therefore should be avoided with other immunosuppressants. Its primary adverse effects include opportunistic infection, infusion reactions, secondary malignancies, and possibly the development of progressive multifocal leukoencephalopathy. Although no cases of PML have been reported with ocrelizumab, in theory, it could lead to the development of this condition. Ocrelizumab is the only agent that is specifically indicated for primary progressive MS. It is also indicated for use in relapsing MS, but this additional indication for primary progressive makes it the only drug in that group. Ocrelizumab is considered a high efficacy DMT. One additional agent I will discuss is used in the treatment of secondary symptoms in MS. These are symptoms that are caused by myelin damage to axons, and they can sometimes be remedied with therapies that have no effect on the underlying pathophysiology. One such agent is dalfampridine, which is a potassium channel blocker. In MS, its mechanism is that it improves conduction in demyelinated axons by delaying repolarization and prolonging action potentials. Essentially, it results in less leakage of potassium out of the neuronal membrane, and therefore delays repolarization and allows action potentials to be more effectively propagated. It is indicated for improving walking in patients with MS, and it can be administered orally. There is a risk of seizures associated with use of this agent, as well as frequent or common development of urinary tract infections. The half-life is about 6 hours, but it is important to note that this half-life is extended by about threefold in individuals with renal impairment, and so some dose adjustment may be needed in those individuals. While dalfampridine is used for the secondary symptom of muscle spasticity, there are other agents that are indicated for some of these other secondary symptoms. Another drug that is approved only in Canada for treatment of muscle spasticity is Sativex, which is a cannabinoid-derived mouth spray that has been shown in clinical trials to reduce muscle spasticity. For bladder dysfunction, the most common agents used are anticholinergics, And you will learn more about these when you get to the incontinence unit. For mood dysfunction, antidepressants are commonly used, and again, we will discuss these in much greater detail in the depression section shortly. For fatigue that is commonly experienced by individuals with MS, stimulants may be used to reduce fatigue. Now is a good time to pause and review everything we've talked about related to the agents that are used in the treatment of MS. One useful way to learn similarities and differences between different agents is to categorize them according to various factors. So, for example, you could categorize the various treatments as exacerbation effective or whether they are a DMT. Drugs that are moderate efficacy versus those that are high efficacy. Oral administration versus parenteral administration. Those that have a more favorable versus those that have a more concerning or problematic safety profile. Those that are small molecules versus those that are biologic drugs. Try to categorize the agents we discussed according to these various factors, and that will help you review the agents prior to MS therapeutics.